Welcome to our lesson on smooth muscle here. In this video, I'm going to sort of introduce you to smooth muscle, a couple of different ways to categorize it, and we'll talk about it in terms of a general physiological introduction. And then in the second video, what I'm going to do is I'm going to talk about the process of excitation, contraction, coupling in smooth muscle, which is quite different from cardiac muscle, and which you've just learned. And in the last video, we'll go over an example of typical smooth muscle and arteriolar smooth muscle, which of course is particularly relevant to the vital fluids block. Okay, so this is smooth muscle, what you see in a microscope, and it is found in the walls of basically hollow internal organs. Okay. Including arterial smooth muscle, which is of course the focus mostly in the cardiorespiratory block. So I went through the three sort of types of muscle at the very beginning of the cardiac muscle lesson. You will remember that cardiac muscle is striated, meaning the myofilaments and the sarcomeres line up so that you can actually see bands as you go from one end to the other in a cardiac muscle cell. This is not striated. You don't see striations here. They just look like regular old cells without that sort of regular structure. And smooth muscle is involuntary. That means that unlike skeletal muscle, where you think here, contract, and your brain sends a message to your skeletal muscle and your muscle contracts in response, smooth muscle contracts in response to signals to your brain that are generated subconsciously. You aren't conscious of it. They're really more visceral in terms of their regulation and less conscious regulation. There isn't a lot of that in smooth muscle. Okay. They do not have a transverse tubular system, so they don't have these invagulations of the cell membrane that go in. They do, however, have a sarcoplasmic reticulum. It is an organelle that is less developed in smooth muscle than it is in cardiac muscle, but it is developed enough to participate in excitation, contraction, coupling, and calcium release, and we will go over that later in the lesson. It is typically arranged in sheets, and it is electrically coupled to varying degrees. So you remember cardiac muscle is connected at the intercalated discs, and you've got gap junctions in there, which are big channels, such that if you generate an action potential in cell 1, it propagates to cell 2, and they act as if they're one cell. There are many types of smooth muscle are electrically coupled where that would happen. Single unit smooth muscle is completely coupled, so that all the cells are, in theory, connected together by gap junctions. So there's a gap junction in the cell membrane of each of these cells that connects to the one next to it, and when one contracts in response to an action potential, all the rest will as well. Okay? This is the most common type. There are many types of single unit smooth muscle are self-excitable, means that there are groups of fibers that generate an action potential spontaneously without any neural input. In that respect, they'll act perhaps a lot like your SA node in the heart, where you're generating an action potential which sets your heart rate. And it's in most visceral organs like the GI tract and so on. That includes the arterioles, which we'll cover in more detail in the final video in this lesson. Okay. Multi-unit smooth muscle, on the other hand, has multiple discrete units, so it has far fewer gap junctions, and all the tissue is not connected together. Instead, more likely you'll have very small groups of cells that are discretely separated and act independently of the groups of cells around them. There'll be small groups. The smaller the group is, the more multi-unit it is. You'll find this in the walls of the larger blood vessels, the airways of the lungs, the iris of the eye. So this is one way to characterize smooth muscle. I wouldn't want you to think that it is a black and white issue, where you can take all smooth muscle and go, well, that's single unit, or all smooth muscle and say, well, that's multi-unit. In fact, most smooth muscle is a mixture of these two, and you don't generally find pure single unit smooth muscle where all the cells are connected. You don't typically find pure multi-unit smooth muscle where there almost none of the cells have gap junctions and none of the cells are connected, okay? So these are really two extremes. What you find more likely in a real sense is a mixture of the two, where in a particular tissue you'll tend more toward one than the other, okay? So I wouldn't want to give you that impression. So this is a figure in your notes that comes later in the lesson, but I'm going to bring it up right now just to quickly show you what multi-unit and single unit look like. So on the left-hand side here, we have single unit muscle, already labeled for you. And you can see these are connected together. So you imagine gap junctions between each of these. So each of these sort of acts as a single unit, okay? On the other hand, you have multi-unit here. You can see these don't have those connections. They don't have the gap junctions. So each one of these acts as an individual unit that reacts independent of the one next to it. So this would be the ideal multi-unit scenario, okay? All right. Another way of categorizing smooth muscle is by the force response that you typically get, the type of response. Tonic smooth muscle is always partially active, so it maintains tone. Remember, we talked about sympathetic and parasympathetic tone and how it was more like volume buttons where you turn up the volume on one side and down on the other, but you never actually turn it down all the way to zero on that other side. There's always some activity, some tone. That would be tonic smooth muscle, always has some tone, and you're either turning up the volume and increasing the contraction, or you're turning down the volume and decreasing the contraction, but there's always some contraction there. It's never completely relaxed, okay? The smooth muscle walls of most blood vessels, the airways of the lungs, various sphincters are like this. They've always got some tone, some contraction in them, all right? Phasic smooth muscle contracts rhythmically or intermittently. You find it in the GI tract, the reproductive tract, the endocrine systems. They usually produce action potentials which initiate a contraction. If either initiate a contraction or increase the contraction, or and therefore increase the contractile response. Okay, usually the more action potential you have, the more response you have in these sorts of muscles. Although that is not always the case, and I'm going to show you that right now. Well, in a moment. We're talking about the contractile response here and increasing and decreasing it. Let's look at what the myofilaments look like. So as in cardiac muscles, smooth muscle does have thick myosin. So it has a thick filament, you can see here in the red. And it does have a thin filament, which you can see here in the blue, okay? But note that the thin filament really just has actin. There's no troponin, there's no tropomyosin. And so because there's no troponin to bind calcium and there's no tropomyosin to regulate the interaction of those myofilaments, as you might guess, the excitation-contraction coupling process is quite different in smooth muscle compared to cardiac muscle. You also have intermediate filaments, which cardiac muscle did not have. These are not contractile elements. They're part of the cell's cytoskeleton, and they're probably responsible for some of the elastic properties of smooth muscle. As you'll see later on, smooth muscle tends to stretch quite easily, and it's really made to operate at many different shapes and sizes, and maybe the intermediate filaments may play a role in that. And then you have dense bodies. So in cardiac muscle, you had Z lines, which anchored the myofilaments. The myofilaments anchored into the Z line and held the actin in place. The actin anchored into it. The actin anchors into dense bodies here. and holds them in place in smooth muscle, and they sort of act sort of like the proteins in the Z lines, but you can see they're much more discrete as they anchor in here. And then dense areas are essentially dense bodies that are on the surface of the cell, and they're involved in mechanical anchoring between neighboring cells. So you can think of these as being like desmosomes were in cardiac muscle. Remember, desmosomes were in the intercalated disc, and they sort of anchored the cell 1 to cell 2. And so dense areas are sort of like that in smooth muscle. Okay, and these filaments are not organized into sarcomeres like cardiac muscle was, and they do not contain myofibrils. Instead, you've got these thin filaments being anchored into the desmosomes here with the thick filaments in between, and they form kind of a diamond shape that you see here in this yellow box. Okay, 1, 2, 3, 4 dense bodies forming a diamond. And then what happens is the muscle contracts, like you see here at the bottom, and the diamond becomes more contracted as well. And that's typically what happens during contraction. Okay, so you have this diamond lattice that contracts in when the smooth muscle contracts. So here's your smooth muscle cell, much less elongated, and that's as your diamonds sort of become contracted down. So excitation, contraction, coupling in smooth muscle. This diagram sort of encapsulates it, although as we're going to find out in a later lesson, how, for instance, the autonomic nervous system feeds into this can get quite complicated, and it unfortunately needs to be taught because much of it is pharmacologically relevant, but we'll cross that bridge when we come to it. For right now, I want you to concentrate just on this, okay? So, contraction in smooth muscle can be calcium dependent, is calcium dependent. So you have calcium that may come from the extracellular space across the sarcolemma, say through a voltage-gated calcium channel, or may come from the SR via IP3 receptors, which are release channels in the SR of some smooth muscle, or calcium-induced calcium release, as you may see in cardiac muscle. You can also see that in smooth muscle where ryanodine receptors, which are release channels, open up and release calcium from the SR. So either or, calcium can rise from either or both of these. Okay. Calcium then binds a molecule called calmodulin. Calmodulin, a very common regulatory molecule that binds calcium and then usually binds to other proteins to have various effects across the cell. So this has targets which it will bind to. One of those targets is myosin light chain kinase. So this is myosin kinase. It's actually myosin light chain kinase, MLCK. You will frequently see it abbreviated as, okay? And when calcium calmodulin interacts with myosin kinase, or myosin light chain kinase, it activates myosin light chain kinase. And when it activates myosin light chain kinase, active myosin light chain kinase uses ATP and generates ADP and phosphorylates. It adds a phosphate to myosin. And when the myosin is phosphorylated, it can bind with actin and cause a contraction. So active myosin light chain kinase means more contraction. And calcium binds to calmodulin and activates myosin light chain kinase, which will cause that phosphorylation, which will allow myosin to bind calcium. And so what you get is formation of cross bridges, similar to what you see in cardiac muscle. Now it's slower than cardiac muscle, but nevertheless it's there, and it actually generates quite a bit more tension than cardiac muscle. So the way that I think of this, and you can safely think of it this way as well, you have a certain amount of energy in ATP. That energy can do one of two things. It can increase the rate at which cross-bridge cycling can take place, and the rate at which muscle can contract, or it can increase the force at which that muscle can contract. And so how that energy is proportioned tells you what that muscle is typically going to do. And in smooth muscle, more of that energy is directed toward generating force, and less of it is directed toward generating a velocity of contraction. So you get slower muscle contraction, but you get muscle contraction with more force per unit cross-sectional area, okay, in smooth muscle. And that all happens due to cross-bridge cycling, which is taking place when this myosin is phosphorylated, okay. Relaxation takes place when calcium is moved back into the SR through an SR calcium pump, or moved back out of the, back across the surface membrane through a calcium pump, or in some cases through a sodium calcium exchanger as a cardiac muscle. But more typically, you will find a calcium pump in the sarcolemma, which will be responsible for that. Another thing related to this is what's called the latch phenomenon. So unlike cardiac muscle, which contracts and relaxes and contracts and relaxes, and which doesn't really has to stay contracted for prolonged periods of time, smooth muscle does. When your vascular smooth muscle contracts and the radius of the arterial goes down and you increase the resistance to flow, it has to stay contracted for a long period of time, as long as the body needs it to, okay? So unlike cardiac muscle, which will just relax again, smooth muscle needs a way to stay contracted without eating up a whole bunch of ATP in the process. So the way it does that is by through what's called the latch phenomenon. And essentially what this is the state of the smooth muscle where ATP is being consumed, but it's being consumed at a low rate. So you have a low rate of cross-bridge cycling. And what that means is that cross-bridges form, that is, the cross-bridge on the myosin binds to the actin, the head binds to the actin, and it stays bound for longer when, when you're in this latch state. Okay, and because it's staying bound for longer, the first thing is you'll utilize less ATP. The second thing is you maintain the tension that you generated for a longer period of time. Okay, and that's what's happening in the latch state. Okay. You also have a phenomenon called stress relaxation, where smooth muscle can be stretched, and initially you have a big increase in tension as you stretch that muscle out. Because the cross bridges are formed, and they're still formed when you stretch the muscle. But what smooth muscle will do that maybe skeletal muscle won't typically do is smooth, or at least it won't do as much, is smooth muscle will eventually, the cross bridges will unbind, and then when they unbind, that stress on the muscle relaxes, and then the cross bridge will rebind again. But it'll rebind in such a way that there's less stress on the muscle. So initially what typically happens is you get a big increase in tension when you stretch the muscle, and then that tension will gradually relax as cross bridges unbind and then bind again, unbind and then bind again. And as that happens, less and less tension is left until you're basically left with the smooth muscle that's essentially back in more or less a relaxed state, or something that's closer to a relaxed state. And so you've got less tension on that muscle. So you increase the tension, and then the tension gradually comes back down. Okay? You have a length-tension relationship in smooth muscle. It is similar to skeletal and cardiac muscle, which we already went through, but it's much broader. So whereas skeletal and cardiac muscle will the tension will go up in cardiac muscle in both over a relatively narrow range, and then in skeletal muscle you'll get past the peak and it actually goes down. Smooth muscle does the same thing, but instead of over this range, it's like this range. It's a much broader range, and you have a much broader range where you actually can generate a fair bit of tension in smooth muscle. Okay? And that actually has a functional role, because smooth muscle typically has to operate over a large range of lengths. It has to be able to stretch and still be functional. So your bladder will go from something like this to something like this. and the smooth muscle in the walls of the bladder still have to be able to function when you're like this, okay? So that means that smooth muscle has to generate tension over a great range. So, you know, the urinary bladder is a really good example. I'm sure that those of you who are female are thinking about childbirth as a typical male. I don't even want to think about that, but you can imagine how important it is for the smooth muscle to be able to operate over a large degree of stretch during a process like that. So smooth muscle, very plastic, very much able to function over a large range of lengths and stretch. All right, that's all I've got for this video. Thank you for your attention.